In this short summary, we'll break down the key differences between PCR and QPCR techniques. There are four main features that set them apart: the PCR mix, the instrumentation, the analysis method, and the type of information you can gain from each. Traditional PCR uses a standard mix of reagents: sample, deoxynucleotide triphosphates, primers, polymerase, buffer, and magnesium ions. QPCR, on the other hand, additionally includes a fluorescent dye or probe to monitor the reaction. Traditional PCR runs on a thermal cycler alone. QPCR requires additional instrumentation, a fluorescence detection system, including a light source and a detector, plus software to read the fluorescence in real time. With traditional PCR, you analyze results at the end, using electrophoresis on an agarose gel. QPCR tracks DNA amplification live, using amplification curves and data analysis software. Traditional PCR tells you whether the target DNA is present or not. QPCR gives you that information too, but also tells you how much DNA is there. To sum up, both PCR and QPCR amplify DNA, but QPCR takes it a step further by measuring amplification in real time using fluorescence, giving you both the presence and the quantity of your target. That's what makes QPCR an essential tool in modern molecular biology. Now you know the difference between PCR and QPCR. Thanks for watching. Before you go, if you found this helpful, hit subscribe, click like, and if you want to take your skills further, check out our self-paced online training QPCR Setup for Success. Find a link in the description.